Mi nombre es Cárcamo Orive, soy investigador Ikerbasque en el Instituto de Investigación Sanitaria Biobizkaia, donde trabajamos investigando los genes que participan en enfermedades cardiometabólicas como la diabetes tipo 2. Sin embargo, yo hice mi tesis en el campo de las células madre y hoy trataremos sobre sus características, aplicaciones y su relación con la búsqueda de la inmortalidad. Kaixo guztioi, zer moduz, ondo? Bai? Egingo dut sarreratxo bat erdaraz, lan egiten dudan lekuari buruz eta niri buruz, eta gero bestea egingo dut ingelesez. Si tenéis cualquier pregunta, en el momento que queráis, simplemente levantar la mano y podéis preguntar cuando queráis, ¿vale? Como ha dicho Sonia, yo ahora mismo trabajo como investigador principal en el Instituto de Investigación. ¿Sí? Yo decía, oh, yeah, sure. So I work in Biobizkaia. That is a research health institute. We have one in Bizkaia, one in Gipuzkoa, and one in Araba. And the thing is that in this institute, that right now, today, it was not like that last year. It has all the different hospitals in Bizkaia collaborating together, so we have around 700 people working or adscribed to the institute, okay? Out of those, around 400, 500 are actually researchers like me, and another 1,000 that are doctors that work in different hospitals and that are also part of the institute. Out of those people working for Biobizkaia, 60% of them are women. We have 7 areas of research, around 50 research groups, 700 research projects, and 600 publications. So what is the mission of the Institute? So the mission of the Institute is actually just to translate whatever we find in basic research to the healthcare system. Okay? It could be just diagnostics, it could be a treatment, it could be a new drug, it could be whatever it will improve the lives of the people. So about me, I just wanted to give you a short story about me. So, as Sonia said, I studied in the University of the Basque Country, then I finished college in the Autonomous University of Barcelona, and from there I came back to San Sebastian where I did my PhD. Okay, so after finishing my PhD, I was recruited to this place that now Sonia has told you. I want to ask you if any of you recognize this place. So this was one of the places I was dreaming with when I was young. So I knew I wanted to be a researcher, and I knew I wanted to work in one of the best places in the world to work for this. So as Sonia said, this is Stanford University. Do you know what it is? What it is? California is very large. It's as large as Spain. So it's close to San Francisco. Okay, it's like 40 kilometers south of San Francisco. The weather is absolutely wonderful in Stanford. The weather is absolutely horrible in San Francisco. So it's a really nice place to do research. And do you know why? Why is this place so important? No? So I am going to give you 3 data. The first one is that in the last 10 years, it has been the second best university in the world. Okay. The second is that they have more Nobel Prizes than most of the countries in Europe, just in the university working right now. And the third one is that just Stanford University has more Olympic medals than the whole country of Spain. So how I ended up working at Stanford, but before going to Stanford, I did my PhD in stem cells, okay? And that is what I am going to talk about today. Before starting, just so you know what we do in Biobizkaia, in my lab. So as Sonia said, we study cardiometabolic disease in a wide range of approaches. So from one side, we want to know what are the genes that actually predisposes you to develop type 2 diabetes or fatty liver or cardiovascular disease. From other side, we want to study what is the beneficial effects of exercise for human health, and we have other venues to try to understand what are those things that either make us sick related to cardiometabolic disease, or that can protect us, like diet, Mediterranean diet versus eating hamburgers every day, you know? So that's what we do currently. But I am going to talk about something that is not so boring as human genetics, genetic variants, and things like that, and I hope you will get interested. So the title of the talk is Stem Cells, Regeneration, and the Quest for Immortality. And as you can see here, there are a lot of topics related with this talk. So you can see that maybe bone marrow transplantation, you have heard about it. It's also related to developing artificial skin. It's associated with personalized medicine, or with arm regeneration, or with other type of things that may sound like pretty incredible, but are actually, as you will see, some of those are actually currently happening. So what I am going to tell you today is why do we need stem cells, what are the main characteristics of stem cells, how many types of stem cells are there, why these stem cells are important, what do scientists use them for, and finally we will associate all that knowledge with why humans die. Okay, so why do we need stem cells? Any guesses? So you can see there that that's much more or less all the different types of living organisms that you can find in the planet. And you have to imagine, and maybe some of you know, that life started as a single cell. Even started before that, but we are not going to go into it. So even before cells, we had other types of more ancestral organisms that were very, very, very simple. But for the sake of brevity, let's start with a cell. A cell is just one cell, right? And that's one thing. Just survive, eat, and duplicate. But as you go evolving, you have animals that are more and more complex. So you need different types of things happening in that organism for it to work, right? So humans are not a cell. Humans have, I don't know how many billion cells. And just imagine that in our bodies we have between 200 and 300 different cell types. Each of those cells does a different thing. You have cells that are in charge of allowing you to see. Some cells allow you to hear. Some cells allow you to transport oxygen in the blood. So that's a lot of different cells. So there is a lot of specialization in the tasks that these cells can perform. And as far as they perform them every day, because you are alive every day, they just get injured or stop working. So you have to replenish that type of pool of cells that do different functions. And for that, we have stem cells. So stem cells are that specific cell type in each of the different tissues, just to start with the concept, that is able to regenerate those cells that you need to work for you to be alive. Okay, so any questions? No? One question, I have one question for you. Do you think that the cells in your body are all the same genetically, or that they are different? How many do think that all the cells in your body are the same? How many people? No? So all of you think that all the cells in your body are different genetically, or what? Please raise hands. Who thinks that all the cells are different? Come on, do you have arms? Okay. So those people will be correct. So you don't even have two cells that are the same in your body. Every single cell in your body has a different genetic sequence. Okay? How's that possible? Because some of the parts of the genome do not do things, and some others do things. And in fact, going back to this evolution is possible because of that, because of having different types of letters in your genome that make the cells act slightly differently is what allows for organisms to evolve. So, what are the main characteristics of stem cells? The two main characteristics of stem cells is that they need to self-renew and they need to be able to differentiate. What does self-renew mean? to generate an exact copy of yourself. And how is possible that they are able to do both things? So just imagine that a stem cell needs to divide and is going to give rise to 2 cells. It has 3 different options. It can generate 2 stem cells. It can generate 2 differentiated cells that are not going to be able to self-renew, or it can generate a differentiated cell and self-renew itself. That is usually what happens. Okay? And why is this important? Why do stem cells need to self-renew? Because in the end, they have to regenerate different types of cells over the course of your lifetime. So now, funny question. This is the half-life of different cells in your body. So erythrocytes are the red cells that carry oxygen. Neutrophils and lymphocytes are defense type of cells that fight the infections that you get. And the platelets are the ones that actually clog your injuries when you are bleeding. And you can see that the median half-life of these cells goes from 10 days to 100 days. So one question. How many kilograms of blood cells do you think you produce per year? And I give you a clue, kilograms. Okay, I will put 3 groups. Who thinks that, okay, I will give you the 3 options and then we will see what people think. More than 5 kilograms, more than 20 kilograms, or more than 50 kilograms? Okay, who goes with more than 50 kilograms? Okay, who goes with more than 5 kilograms? So the 4 most clever people in the room, and I will explain why. So more than 20 kilograms? Okay, so if you give the answer of 5 kilograms, you are safer than giving the 20 or the 50. So you will be right no matter what. Okay, that was a little trick. But yes, it's 26 kilograms. So each of you, every year produces 25 kilograms of new cells. So imagine the power of stem cells in your body. Okay, so it's pretty amazing when I got to see this number, just to think the number of times a cell has to divide to produce 26 kilograms, well, a table like this, maybe, 26 kilograms of cells, okay? So, another thing is, as I was telling you, in the blood, we have many different cell types. I talk about red blood cells, white blood cells, but you can see here, for example, on top, you have a hematopoietic stem cell. And that hematopoietic stem cell has to give rise to all these different cell types, right? And how does that? So initially it divides, generates a more differentiated cell and self-renews. And once it self-renews, that's it. The cells that has generated then goes on to a differentiation path that gives rise to all these different cells in your blood. So it's not just that it's able to give rise to a lot of cells, and is also able to give rise to a lot of different cells, okay? So how many types of stem cells are there? So mainly, and we will see now why, there are 3 main stem cell types. Some that are embryonic stem cells, some that are fetal stem cells, and some that are adult stem cells. Anyone wants to guess what's the difference between those? And it's associated with what we just saw. No guesses? It's about potency. So the embryonic stem cells are pluripotent. What does that mean? It means that it can give rise to any cell type in your body, which means that embryonic stem cells can give rise to blood cells, nerve cells, neurons, bone, whatever you want. But adult stem cells and fetal stem cells are not pluripotent, are either multipotent or unipotent. What does that mean? So we have some stem cells, and we will see a picture later, that are able to give rise to adipocytes, skeletal muscle cells, bone, or cartilage. And that means that they are multipotent. They can give rise to more than one cell type. And there are other adult stem cells that give rise to just one type of stem cell to one type of differentiated cell, and those will be unipotent. Okay? So now, there was a lot of ethical discussion about the use of embryonic stem cells, and the good thing is that nowadays we don't care anymore. And why we do not care? Because now we are able to produce artificial pluripotent stem cells. They are called induced pluripotent stem cells, and the only thing that you need for that is a bit of blood. So we can draw blood from a person, we are able to reprogram those cells, they go back in time, and they are able to behave like embryonic stem cells. And why is that important for us? Because then we can generate any cell type that we want to do research. And we don't have to use embryos anymore. So there is no need to use human embryos to generate embryonic stem cells. We can generate artificial embryonic stem cells from the blood. So there is no ethical concern there, because in the end it's just the same thing when you go to a hospital, they take the blood, we reprogram them, and then you can generate any cell type that you want. Okay, why are stem cells important? So they are important for development. So we have talked that they give rise, or they are needed to have many different animals. We have talked that they are needed to have many different cell types, and just that you start with one cell, okay? So when you have the egg and the spermatozoid, they just merge and they give rise to one cell. And from that cell, 9 months later, okay, is a baby coming out. And that baby has a lot of different differentiated cells, a lot of cells doing a lot of different things. So it's really critical, of course, the function of stem cells during our own development. From one cell here, you go dividing, until that stage, or between this stage and this stage, the cells are totipotent, meaning that theoretically you could get a human being from each of those cells. So each of those cells are able to generate a human being. At this stage, if you take out this part of the blastocyst, you can generate embryonic stem cells, and later on we lose that capacity. Okay, with one exception that we will see a bit later. So as we said, it's very important, the stem cells, because you have to generate from one cell all those different cells that we have inside. Okay, without the stem cells, there will be no animals, no human beings, maybe some bacteria, and that's it. So why? Another thing that they are important. They are really, really important in this dichotomy, in aging versus cancer. They are two sides of the same coin. Okay. So, do you remember Benjamin Button, the film? Have you seen it? Maybe no. Maybe you are too young. It's Brad Pitt, so it's a pretty known film. Basically, that film, what it does is showing a person that ages really, really, really quickly. Okay, and there are other films that show you people that never age, like vampires, right? So, what is this thing about aging and cancer? So it's something that is associated with stem cells in tissue homeostasis. So if the cells, or the stem cells, tend, let's say, to simplify a lot to self-renew, and do not differentiate, the likelihood of developing a cancer is very high. On the other side, if the stem cells cannot regenerate themselves, most likely what is going to happen is aging at a faster rate than regular people, and we'll see a couple of examples later. Just imagine that in cancer, nowadays, we know that there is a cancer stem cell. So tumors, actually, to be able to maintain themselves, usually they have a stem cell population that has mutated, that doesn't work as it should be, and that its function is to actually regenerate the tumor. That's why in many cases, there is people that has a treatment, the doctors say, oh, you are free of the tumor. And suddenly, 5 years later, the tumor is back. And the reason is these stem cells that reside in the tumor. Because nowadays, we are not able to locate one tumor cell. We are able to locate a mass. But whenever the mass is gone, it could be that there are some stem cells that are from the tumor that reside and that proliferate. So there are a lot of groups researching on how we can exterminate these tumor stem cells. Okay, so regeneration. Who thinks there is an animal that can regenerate the tail? Solo? You haven't never seen a... joder. ¿Lagartija? Yes. Okay. Do you think there is any animal that can regenerate an arm? Yes? Okay. And the heart? Yes? Okay, so you're right. So actually this salamander, that is an oxalot, originary of some dams of Mexico, is able to regenerate the limbs, the heart, the tail, the brain, the eyes, the kidney, and the spinal cord through life. So basically, can you regenerate any part of the body? So in the animal kingdom, we have examples of animals that are able to regenerate any part of the body. It doesn't happen with us, okay? We know what are the limitations of our regeneration. But in the case of this animal, what happens, and is very interesting from the standpoint of studying stem cells, is that whenever one of these animals, it can be this newt or a salamander, gets the limb amputated, those cells that are in the limb are differentiated cells. It's bone cells, it's blood cells, it's skin cells, but we don't know why. There are some animals that are able, when that injury happens, those cells that are in that injury area do the same as we do in the lab. They go back in time. They differentiate, they stop being a bone cell, they stop being a skin cell, and they go back in time. And they behave like if they were, let's say, embryonic stem cells or stem cells. And from there, they are able to newly generate bone, blood vessels, and everything. And you can see here, around 70 days later, the newt that didn't have the leg, they grow a new arm, a new leg. Okay, so it's pretty amazing, the case of these animals. And that means actually it's something that has been lost through the evolution. So there were animals, especially animals that live in the water, that had a lot of capabilities to regenerate that we have lost over the evolution. So what do scientists use them for? So transplants. I don't know if some of you maybe have heard about bone marrow transplantation, trasplante de médula. So that was the most common type of use of stem cells. So someone got a leukemia or a myeloma or different types of blood cancers. And what the scientists or the doctors will do is you eliminate the blood from that patient. So you irradiate the patient, so there are no blood cells from that patient, in theory. That's why the people that go through these processes are pretty tired and have that type of thing. No? And then what you do is you transplant a bone marrow. And why is important the bone marrow? Because in the bone marrow is where the hematopoietic stem cells reside. So what you are doing is eliminating the blood, introducing stem cells from the same or a different person, and it regenerates the blood completely and hopefully free of the tumor. Nowadays, that has been mostly changed, and nowadays the thing that is most used is umbilical cord blood. So maybe you have heard in the news that there are banks for umbilical cord blood. And why is that important? Because the umbilical cord blood from the newborns, it has a lot of stem cells. And it is easier, let's say, to use than the matching of a bone marrow transplantation. So nowadays, most of the transplants are done with umbilical cord blood. Okay? So this is one case that has been used for the last, I will say, 50 to 70 years. So we didn't know stem cells existed, and this was already happening because we know there was something in the blood that was able to regenerate the blood. And it was actually a researcher from Stanford University, Irving Weisman, who was the first one trying to describe the hematopoietic stem cells and the hematopoietic stem cells in the tumors. So what other things can we do with them? So we can generate artificial tissues. Let's see if someone can guess what this is. When you know, just yell it, Say, what do you think is this? No? No one? Venga, valientes. What is that? Do you know to read? Okay. So what is that? It's heart, right? Wow, that's heart in a petri dish. So now I have two hearts. One in the lab and one here. So I will put it again, and please, someone tell me what is wrong with these cardiac cells. Anyone? What is wrong with these cells? What is that they should do that they are not doing? So, yes, much more or less that. They are not moving at the same time. That's right. So our hearts, all the heart pumps blood at the same time. In fact, when all the heart is not pumping at the same time, you have a problem. You can have serious problems if your heart is pumping like pa 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 pa. If you see again, not all the cells are beating at the same time, okay? And that's one of the biggest issues to actually generate cardiac tissue nowadays. So we are able to generate these cardiac cells, but we are not able to make them beat all at the same time. And the second problem, do you know what it is? The size. It's very difficult to make the organs the size they should be. So you can generate a lot of cardiac cells, but they have to have a specific size and a specific conformation. So for example, one of my colleagues in Stanford, what they used to do, or one of the research projects they had, was to take hearts, human hearts from people that have died or that they were transplanted, and they will eliminate all the cells from the heart. You will have the scaffold, the actual form of the heart, and they will put cells on top of that scaffold to try to see how to generate a new heart out of that one that was decellularized. Okay, so, and lastly, in fact, when I was doing my PhD in San Sebastian, we were some of the first ones in Europe, I will put it this way, to generate artificial skin for the butterfly kids. I don't know if you know about the butterfly kids, but they have mutations in the collagen genes, and basically if you touch them, the skin just peels. It's a pretty horrendous disease, and that has no cure, but we were one of the first labs to generate artificial skin in the laboratory. So we could generate actually your skin, like your skin, in the lab, you take it and you put it like if it was una tirita, like that. There are other things that can be done. For example, now there are pretty successful clinical assays with regenerating macular degeneration and other types of regeneration for different diseases. Okay, cloning. Who knows the name of this sheep? What is the name? Shout it. What is the name? Come on. No puede ser. What is the name? Why is important that sheep? Dolly, yes. That's Dolly. So Dolly was the first mammal cloned. So you can see the original animal and the cloned animal. Basically, we will see now how cloning happens. But this happened around the year 90, I don't remember, 1995, 6 or something like. And it was the first mammal to be cloned. And it was cloned by Ian Wilmuth, that I have the pleasure to meet. And I want to give you a fun fact about him. So he was the first scientist to clone a mammal. And when I saw him in a conference, he will go up to the stand to give the talk with shorts, sandals, and white socks. And I was like, okay, if you clone an animal, you can dress as you wish, because it was pretty terrible. Another question. Okay, the dolly was the first mammal to be cloned, but what was the first animal to be cloned? Come on, guess, say animals. Which animal? Say animals, please. Any animal. Come on, more animals, more animals. Frog, 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 I heard frog. It was not exactly a frog, it was a toad, un sapo. So that was the first animal to be cloned. And in fact, the guy that cloned it was John Gurdon. And John Gurdon was awarded the Nobel Prize for those work. So in fact, who cloned Dolly didn't get the Nobel Prize. It was the person that actually discovered the technology to clone animals. Not because of cloning animals, but because it allowed us to study how reprogramming, how stem cells work. That same year, also Shinya Yamanaka got the Nobel Prize because of the discovery of induced pluripotent stem cells. So the stem cell field has been really, really, really important. In fact, I remember when I was in Stanford in San Francisco, I saw them for the first time in public because they were giving a conference in San Francisco and they were awarded the Nobel Prize while they were actually at Stanford. So pretty amazing to know them in person. Okay, so how does cloning happen? There is like, I believe is 41 people, 33 women, 8 men in the room. So for this, the 8 men, useless, absolutely useless. With men, there will be no cloning. Why? Because there is a fun thing about the egg. Do you know what it is? So the eggs, or los óvulos, just to understand each other, they have the capability of reprogramming an adult nucleus. So when you take an adult cell, take the nucleus of the cell, and you put it inside an egg, the egg is so powerful that it's able to reprogram that nucleus and make it behave as if it was just a newborn, and is able to generate a new cloned animal. So without women, there will be no possibility of this type of regenerative capacity. Okay? Yes? Questions?
So for the moment, there wasn't any male animal.
No, it doesn't matter. You can generate a male or a female because that will be determined in what is the adult nucleus you take. So if you take a male adult nucleus, it will be a male. If you take a female adult nucleus, it will be a female adult nucleus. And so you know, we have cloned almost any animal in the world, but not primates. Not orangutan, not gorilla. And we don't know why, but there is a jump between, for example, monkeys and primates. There is something in the especially epigenetic regulation of these cells that doesn't allow us to clone primates. There are no cloned people around the world, okay? No matter what you hear, most likely, unless there is a secret agency doing nasty things, most likely there is no cloned people around the world, okay? We can also do disease models. And why is this important? Just imagine one thing, Alzheimer, Parkinson. Where does it happen? In the brain. I promise you that people yield a lot if you try to take a brain piece. I was kidding. So it's just that it is very difficult to study some sorts of diseases because of the accessibility. For example, studying diseases that are associated with a fat is pretty easy because there is a lot of people that goes to a clinic and says, I want bariatric surgery because I want to lose weight. So we can get chunks of fat tissue quite easily. But muscle is not so easy, and especially neurons is not easy. So how on earth are we going to study Alzheimer if we cannot get cells to study what happens to the cells? So I give you some hints before. So you remember the induced pluripotent stem cells? I told you we can take a blood sample, reprogram the cells, and then generate any cell type in the body. So what you can do is, okay, you have healthy people and you have people with Alzheimer. You take blood samples from those people and you generate induced pluripotent stem cells that are either coming from a patient that has the disease you want to study or that they are healthy. Then, from these cells, you can generate neurons, and you can compare what happens between the neurons. Okay? That's why doing disease models is so important, because otherwise there are diseases we could not even study. Okay, animal models. So we are able, for example, to generate fluorescent animals. And why is that important? Why is fluorescence important? For research. Because that allows you, louder, exactly, to see them. So for example, imagine that we want to see what happens with human cells in the brain. And you, for that, need a brain. And it's not allowed nowadays to put human cells into another human. So what we do is to put those human cells into a mouse. But how do you distinguish the cells? For example, you can make fluorescent cells. So you will distinguish the human fluorescent cells from the mouse cells. A silly example, okay? In fact, I believe even there are some countries that actually commercialize some of these animals. So I believe in Japan they actually sell fluorescent fish. So if you want to have a really fancy fish type of thing at home, it's something you can do. But in theory, it's also really helpful for research, okay? And then artificial meat. Who likes burgers? A lot of people, I am sure. Don't eat burgers, guys. Or eat as few as possible, if you can. Nowadays it's known that it confers as much risk as smoking for future cardiovascular disease 40 years from now. So processed meats are not as bad as smoking, but close. Okay, so you know about it. By the way, I know no one is going to take care of what I said, because one of the things that we have seen in the studies is that people eat what they want, no matter what you tell them. You can tell them, if you do this, you are going to die. They will continue doing it. So one thing that nowadays we do is that also the body mass index, that is actually the weight, is associated with genetic variants that act through, we thought it was going to be the fat tissue. So you have a genetic variants that do something in the fat. Now, genetic variants that act in the neurons. So for the main population, in general, for the population, most of the genetic variants that are going to determine what your weight is are associated with your behavior, with your decisions, with what you choose to eat. Okay? Okay, artificial meat. What is this? Basically, we can do hamburgers in the lab. So you can take stem cells, muscle stem cells from a cow, you culture them in the lab, and then you get hamburgers without killing a cow. And actually, there is a company that is doing this in San Sebastián, in Parque Tecnológico de Miramón. There is a company actually trying to commercialize this. Yes?
Can you create red meat with the same nutrients that?
Good question. I don't know. I am guessing that it's going to be not exactly the same, because in the hamburgers you have more things than actually just muscle cells. You have blood, you have other things. So I'm not sure what is the stage of development of the product, but I'm guessing that maybe they will do a bit of a mixture. Maybe they are going to try to do something mixing the actual stem cells from an animal or muscle stem cells from an animal, maybe adding other types of proteins that are maybe from plants, or I am not exactly sure, but you're right. I mean, the muscle has a lot of different things. Muscle has muscle cells, blood cells, fat cells, a lot of different things. So we'll see how that works, but actually, let's say that it's pretty amazing that it's able to generate something similar to a hamburger in the lab. Okay. So a brief summary before going to the last part of the talk. So we have seen that stem cells are necessary to sustain multicellular organisms. They need to self-renew and differentiate. And they have different potentials depending the human developmental stage they come from. They are important for development, aging, cancer, regeneration, transplants, artificial tissues, cloning, disease models, animal models, and artificial meat, for example, just to give you some examples. There are more examples about the uses of stem cells, okay? So now, just a couple of silly pictures, because I am guessing maybe one or two of you are thinking, how do they look like, right? So, guess, what type of stem cells are those balls? Which tissue? Where do you think they are? Just guess, randomly. Skin, bone, brain. They are blood hematopoietic stem cells. So these small balls are the stem cells that give rise to those 26 kilograms of cells per year. Those balls over there. And do you know what the other ones are? These flat cells. Those usually are mesenchymal stem cells. So the mesenchymal stem cells, apart from generating bone, generating cartilage, and generating adipocytes, they also give support, or part of the support, the hematopoietic stem cells need. So it's a work in progress between both stem cells, just for the hematopoietic stem cells to have a house where they can self-renew, as we were discussing. Okay, these ones. What is this? More. What do you think? Almost. You are very close, eh? So this type of cell can give rise to muscle, can give rise to bone, but it's the other one. This is fat. This is fat cells, okay? So these cells are mesenchymal stem cells that I actually culture with my hands, that I put to differentiate, and initially they look like this. The fat has really small balls, but if you leave them, they go fusing, and in theory, our adipocytes, our fat cells, are just one single droplet of fat, okay? One in each cell. More likely like these ones you can see here, okay? So they exist, you can take pictures of stem cells. It's not that I talk about them and they are something. You can actually see them, you can mark them, you can isolate them, you can do a lot of things with them. Okay, why do humans die? Oh, mama. So we are going to try to relate what we have seen about the stem cells and how they help, or they could help us related to the different causes of death for humans. So, 4 main reasons. Starvation. Of course, if you don't eat, you die, right? So we have seen that with the stem cells we can generate food. We can actually generate burgers. We can actually generate things that will not require animals. Okay? Injury. Of course, I mean, get in a really serious accident is one of the main causes of death nowadays in the world. Or you can get serious injuries, like we have seen people in a wheelchair and things like that. And stem cells actually can help with that. In fact, there is starting to be some cases where some approaches are hopeful to be able to get people standing from a wheelchair and walking again. It is not happening yet. It has happened with some gorillas, for example. It's promising, and the problem there is not so much that we cannot generate neurons, because we can generate new neurons to replenish what is. The problem is that usually they don't go where they should go. So imagine that someone has a broken spinal cord, so there is a gap. And that's why they don't walk, because the neurons from the legs do not communicate with the neurons from the upper part. So you need to make them contact. So when we try to put the stem cells in here to generate new neurons, the neurons start to grow like this. Plop, 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 plop. Or plop, 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 plop. So nowadays, it's working better that they are starting to grow the direction they should. So many things with biology is not just, I am able to do something, but I am able to do it the right way. Okay? So stem cells can help you regenerate. Of course, we have seen the case of the newt, the salamander. There is also a fish that can regenerate the heart. It's very helpful to study different types of things in the lab. Okay, and then disease. But disease is not just one type of disease. We can have a disease that is environmental. Imagine the Chernobyl kids. They got sick, but they didn't have any genetic disease. It was that they were exposed to radiation, or you can be exposed to a chemical agent, or you can be exposed to different things that actually will generate a disease, right? And actually, it can also be that you get some sort of infection. cholera, typhus, tetanus, tuberculosis, right? The thing is that for those things, stem cells are also very important, because if you want to study a virus, how do you do it? So to generate enough cells that you are able to actually produce, for example, viruses to study, you can use stem cells. Or you can use stem cells to see what happens if you are exposed to radiation. You have cells in the culture, you expose those cells to radiation and see what happens when you generate blood. One question, does anyone know where the vaccine for the flu, para la gripe, is produced normally? What do we use to produce flu vaccines? No? No one has heard it? That's what you produce from there. But usually what you need comes from a thing that does kikiriki. So usually the flu vaccines are produced in eggs. That's why it takes a long time to produce the vaccines. And that's why every year you have to predict what's going to happen next year. So usually the vaccines have to start being produced 1 year before. Just remember what happened with COVID. Why we didn't have a vaccine for months? Because we need to produce them. The actual advance of the COVID vaccines was that you don't need the eggs anymore, because they are RNA vaccines. So you can produce those messengers in the lab without the need of eggs or anything. Okay. And then, of course, you can have genetic diseases, and there stem cells also help, because the stem cell is going to have that mutation or that variation that is conferring you the disease. And as we said, we can generate any cell type in the body. So you can study, for example, which genetic variants are going to predispose you to develop Alzheimer's or diabetes, studying the cells that derive from those stem cells. And then aging. Of course, we already saw that if our stem cells will be immortal, we will be immortal. Let's put it this way. So what happens is that the stem cells have to divide. Okay? We have seen that they need to divide in order to produce, for example, a lot of blood cells. And one of the issues is that we are programmed to die. And the reason we are programmed to die is to liberate resources for our offspring. So the nature thinks that if you get kids, the kids get kids, the kids get kids. In the end, there is no food and all die. So we are programmed as animals. From one side, it's not the whole story, but for you to imagine that once you get an offspring, you are dependable. And for that, there is a mechanism that is called telomere shortening. And the telomere is just a thing, a cap that the chromosomes have. Okay? But each time a cell divides, the cap is being cut. And there is a moment after a lot of divisions, let's say 40, 50, 60 years of divisions, that the cap is gone. And some genes activate that actually activate aging. Actually, currently, we know that there are 3 stages of aging. One that is happening around my age, oh mama. One that happens around 60 years old, and then one that happens around 75 years old. And those are established time points in humans nowadays that start different stages of the age. And of course, stem cells are critical to study those processes. Okay, now 3 final slides with trivia. And please try to guess. Which of these animals lives longer? Okay, think 5 seconds. Okay, think 5 seconds, think 5 seconds. Okay. Ready? And please, raise your hand. Who thinks it's the turtle? Okay. Who thinks it's the lobster? No one. Poor lobster. Who thinks it's the elephant? No one? And who thinks it's the jellyfish? Yeah! Actually, the jellyfish is immortal. This jellyfish can live forever. Forever. Seriously, forever. So I am going to explain why, but I'm going to start with the lobster. The second animal that could live longer is the lobster. Theoretically, biologically, the lobster is also immortal. But it's actually not immortal. It actually dies, but not because of biological death. Lobsters have one thing that we have just talked about that is associated with what we have talked. They have an active telomerase. And what is the telomerase? The telomerase is an enzyme, a protein, that whenever the cells divide and they lose that cap, that makes us age, they are able to replenish that gap. So in lobsters, their chromosomes do not age. In theory, biologically, lobsters do not age, but they die. Does someone know how they die? Most of them, apart from being eaten by us or being eaten by a shark, okay? I mean, what is the reason why they will naturally die? Could be, that's true, could be, but it's not the answer, the exact answer I am looking for. So think about the parts of the animal. They have two main different parts, right? Go ahead. No, but you're going closer. So the lobsters have a soft part that is the one we like. Anyone likes lobster here? Yes? No? And it has a harder part that is the outer part. So while the body, that is the soft part, grows, the outer part, that is the, I don't know the English word, the shell, has to also grow. But at some point, it doesn't happen at the same time. So the body starts to grow faster than the actual lobster can grow the shell. And what usually they die suffocated by their own self, because the cell cannot grow fast as the body. So there is a moment where the poor lobster does not enter their own self, okay? But they are biologically immortal, much more or less. So then let's see the case of this jellyfish. This jellyfish is called Turritopsis dorni, and you can find it in the Mediterranean Sea. So if you go on vacation to Benidorm, maybe you will see one, and they are immortal. Do you know why they are immortal? Because they are smarter than us. And how is it possible that a jellyfish is smarter than humans? Because they make the right choices for them. So this jellyfish, when it is an adult, is living in the sea. If they find there is no food, if they find there is a contaminant or some sort of toxins, if they find the companies they have around are not good for them, they can reverse to an egg stage. So the jellyfish goes back in time, and is an egg. And it can stay in that egg stage until the bad companies disappear, let's say animals that want to eat her, or that the toxin is clear, or that there is newly food, and they can develop again from that egg stage, blah, 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 to an adult stage. How many days can they do that? Theoretically, forever. Just think about ourselves. Usually, we don't care about toxins. We smoke, we drink. We don't care about companies. You have that toxic friend, but you still go with them, right? And then food. We talked about hamburgers before. So I believe the jellyfish is much smarter than we are, okay? There are other examples of few examples of immortal animals, but theoretically, biologically, it's something that is possible. Okay. Now a couple of examples in the human race about immortals. Do you know any immortals? You are too young, but I like this picture. I love this film. Have any of you seen Christopher Lambert in The Immortals? Yes. Do you remember how they die? Exactly. That will kill an immortal and a not immortal. But yes, that was a film that was pretty not scientific, okay? But the idea of immortality is something that for the human race has been always something that is a goal, let's say. And for what we know nowadays, is a goal that is achievable in the long term. So it's not something that is crazy. Or it's possible that we will live long enough that you will not distinguish from immortality, because we know many of the mechanisms that actually regulate aging. And most of those mechanisms are also associated with human disease. Just imagine 2,000 years ago, no one got Alzheimer. No one. Why? Because they didn't grow old enough to develop Alzheimer. So it's just because we are growing over 55, 60 years old that now Alzheimer is a problem, okay? So both the response to immortality, for example, and the response to why Alzheimer happens usually is associated. And just a clue about that. we now know that most of those genes that now harm us, that predisposes us to diabetes, predisposes us to Alzheimer, they had a protective effect maybe 20,000 years ago. So the same genes that now are harmful, maybe they were a good gene to have or a good variant to have when you didn't have food. So those variants were good to survive when there is no food around. But nowadays, as we said, we have plenty of food. So all those variants nowadays that we can eat whatever we want are actually usually the variants that will harm us. And last, maybe you are too young too. There is a case of a human being that is immortal, maybe two. But I like this one. Have you ever seen Saber y Ganar? Yes? Yes. So this is, I believe, one of the few examples of a human being that is immortal, because he has been like with this program for 40 years, and he actually has been the same for 3 decades, Jordi Hurtado. It's a fun fact. If you look internet, there are a lot of memes about him, a lot. I mean, he's the type of person that has never changed. And then we have another one. I don't have a picture of him, but I think there is another guy that may be immortal. That is Julio Iglesias. He's still alive, right? I mean, I believe he has to have 250 years or something alike. So of course I am kidding. But it is true that depending, and you can see around you, I am sure you have an uncle, an aunt, or a grandmother that you say, everyone tells them, oh, you look so young. And we have a lot of examples of people that look like that. And in fact, it's one of the projects that I will want to do in the future that is called the elderly. And the elderly are those people that are over 80, 85 that never had a disease. Yes, there is people that is 90 years old that never had diabetes, never had cancer, never had Alzheimer, never had any type of disease. And usually those people are the ones that are more active, are the ones that are younger or look younger. So it is all related to each other, okay? So with this, please, any question, okay? Whatever you want, ask whatever you want. There are no silly questions, there are silly answers, okay?
It's like a too hypothetical question, but in the case that we can't generate ourselves, it can be that we are going to be able to duplicate ourselves, no?
I mean, human cloning is a near-term possibility. I believe that cloning a human being technically is going to be possible in the next decade, 20 years, I don't know. A different thing is if that's going to happen or not. Usually when we are able to do something, humans, we usually do it. So we invent the atomic bomb and someone decides it's a good idea to throw it over other humans. So if cloning is possible at some point, who knows?
But for example, if you clone, you are cloning everything, but for example, the memories and everything will clone.
No, no, because, and this is important to understand, what you are cloning, and it's not even exactly the same, because remember what I told you, you don't have 2 cells that are the same in your body, but you start with 1 cell. How is that even possible? So that cell has the same genome, but then when you are formed, none of your cells is equally genetically. Why is that? Because nature has prepared our genome to make errors. So whenever a cell divides, just imagine that our genome has 3,000 million letters. And each of those 3,000 million letters has to be replicated each time a cell divides. And usually there is an error approximately 1 letter out of each 50,000 letters. So every time a cell divides, you incorporate maybe 400 letters or 4,000 or 40,000 letters that are new and different from the previous cell. And that's important for evolution, for new functions. Some of them are going to be bad and some of them are going to be good. So even if you clone something, that cells that are going to be dividing from the original entity are going to be different. So it's going to be very similar because the genome in general is going to be the same, but not all the genome is going to be the same. And second, we know that environment makes a huge difference, especially in our behavior. Okay? It's not the same to have nice parents than to have strict parents, than to have parents that never care about you, or the family or the environment. That's important too. It's not that it determines who you are going to be, but it influences it. In the end, it's your choice what you want to be. But of course, we know that how the people are. Maybe you have friends that are twins. In my case, I have friends that are twins. And we call one the good, and we call the other the bad. So imagine, and they are twins. Exactly the same genetic makeup.
So there is not going to be any exact correlation.
That's very, no, there's, a ver. Okay, 99.9, yes, it's going to be the same. But out of 3,000 million letters are some that are going to be different. By random chance, it could happen that one of those letters that changes is in a specific protein. Imagine, the protein that determines if you are smart. That doesn't exist, but just for the ease of thinking. And then suddenly you have a smarter person that is exactly the same as the previous person. That's not going to happen, but it could happen some sort of small changes. So even if you clone a human being, they are going to be slightly different, for sure. I don't know who was next.
You said before that the human chromosome has a little cap that disappears with age when they reproduce themselves once again and again. So if we can produce that hat in a human chromosome, like in the lobster, that doesn't disappear, a human can be immortal.
So now let's say that we are here. We say that if the cells divide, you age. And what you are saying, oh, let's make the telomeres being repaired constantly so we don't age. Yeah. Do you know which cells do that? Okay. So there is a balance. It's one of the things you will need. You will need the telomeres to be repaired. But if you have an active telomerase, that's, for example, a hallmark of tumors. Tumors usually reactivate telomerase or do other things that allow the cells not to get the telomeres shortened. So that's why this slide was important in the sense that there is a balance between aging and cancer.
Will we be able to enhance skills like intelligence or speed or strength with stem cells?
Okay, I will start putting a couple of steps back. No? One of the projects I am nowadays involved with Stanford University, we are looking for top of the top professional athletes in the world. Okay? Because we are interested in understanding which are the genes or the genetic variants that allow these people, like Kilian Jornet or Pogacar or other sportsmen that you can think about, to have those capabilities. And my interest is because I believe that some of those genes that are actually give that capability are also associated with cardiometabolic disease. Because we know that exercise, listen, exercise is the best treatment possible for diabetes, fatty liver, heart attacks. If you want to decrease your risk of being sick in the future, exercise is better than any pill available. We don't know why. We don't know why exercise is good. We have some hints, but we don't actually know the mechanisms. Okay, I will start with that. Then, what we know is that, for example, hate is the human trait that is more genetic. I talk about genetics versus environment. So hate, how high you are, is 95% genetic. But nowadays we are taller than our grandpas. Yes, it's 95 genetic, but if you don't eat enough, if you're in the Second World War, it doesn't matter what your genetics are. Yes, you are going to be the tallest one, but shorter. Okay, second point. So, without knowing which genes actually give the athletic capabilities, without knowing exactly what are the environmental cues that affect those type of traits we were talking about, it's difficult that we are going to use anything for anything. And this is important for all the different diseases. If we don't understand the cause, we are not going to have a treatment. So it will be possible. Bueno, you have some examples. So for example, Rafa Nadal. ¿Vale? It's not that it's enhancing its capabilities, but he had a lot of problems with the knees over his career. And in fact, the treatments that he usually has had, it has been factors that are secreted by stem cells. So there was a research lab that discovered that the stem cells secrete a combination of growth factors that actually make the knees to heal better. So yes, it has an application for health, for being better. But I would say we are many, many years away from being able to use stem cells for anything like that. First, we need to know why. And once we know why, we should go and watch for a treatment. That is something that nowadays doesn't happen so often with funding agencies, because usually the governments or entities, when they give you money, they want a cure. They don't want an answer. And I believe we should start to shift that paradigm. You're not going to cure Alzheimer's tomorrow. First, you need to understand why Alzheimer happens. We know what happens once you get the disease. But for example, 60 years later, we don't still know why Alzheimer happens. No matter what they say, we don't know exactly why it happens. So first, we need to know why it happens, and then we will try to find a cure or any application for stem cells that you can dream of.
Thank you.
Any other question?
Who wants to be immortal? Anyone wants to be immortal? No? Good choice. Good choice. You will get bored.
So I think we can finish now. So you can go away for the weekend.
And if you want to ask any questions, I mean, after we finish, you are more than welcome, okay?
Okay, thank you, Ivan.
Thank you.
For this talk, and thank you to all of you to be here today. So